This protocol enables researchers to analyze bacteria grown under conditions that more closely mimic those found in a mammalian host, which are metal-limited through a process called nutritional immunity. This protocol facilitates generation of consistent, reproducible bacterial growth under metal-restricted conditions and can be adapted for growth of many different types of bacteria.
2 days before beginning the experiment, streak gonococci from freezer stocks onto GC medium plates for a no more than 24-hour incubation at 37 degrees Celsius. 14 to 16 hours before the growth experiment, streak single colonies onto fresh GC medium plates. On the day of the experiment, add 5 to 10 milliliters CDM to an acid-washed 125 milliliter baffled sidearm flask and use this medium to blank a clet colorimeter. Use a sterile cotton tip swab to inoculate 20 clet units of CDM from healthy single colonies.
Generation of a healthy Neisseria gonorrhoeae inoculum for subculture into the 96 well plates for liquid growth assays is critical to the success of all downstream experiments.
Incubate the cultures at 37 degrees Celsius and 250 revolutions per minute for 1 to 2 hours until approximately 1 mass doubling before diluting the cultures with a sufficient volume of CDM to reach half of the initial culture density. Then return the culture to the shaking incubator. To prepare metal-loaded proteins, add furation solution to a 10 mg per milliliter human transferrin solution.